Welcome back. We had left off with split reporters, soluble in membrane, and I think I left with this slide. The yeast 2 hybrid and the split reporters are prone to limitations, and I know we saw that in the yeast 2 hybrid for sure. We had colonies that were weakly growing, so there was an indication of a weak interaction. So we have no interaction, strong interaction, and weak. What does that mean, weak? Uh, it could mean a lot. It could mean it's unrelated by chance, or it could be saying something. It's really hard when you go through that for a used to hybrid to weed through. You get stuck in the swamp quick. So usually maybe use that as a way to set up certainly the strong interactors and then move forward, keeping the weak ones on the shelf until you can get back to them. So informatics is leading the way on characterizing the obvious, the ones, the low-hanging fruit. The challenge for bioinformatics is going to be finding these weak interactions, these rare interactions, the unknown unknowns, and that's going to take some time. The synthetic biologists need to understand all aspects of the interactions, not just the obvious ones. And so we're still a little bit away from characterizing all that information, but it's coming. So, okay, so there are limitations. And so we turn to other interaction technologies. If you remember our chromatography, we could do affinity chromatography. We could actually take a bait protein and pour over a cell extract under non-denaturing, so no SDS conditions, to then isolate proteins that bind to it under the conditions we can modify. And again, things like salt, pH, temperature, there's a lot of variations and variables there, but that's one way we could do it, kind of, and then each of the proteins can then be through Edmond degradation or the Maltitolf electron spray, charge the mass protein sequencing and fingerprint interactions that we can do. So that's one way. Uh, Things like a Western blot, and there's something called a far Western that we'll talk about. So a far Western, co-immunoprecipitation. So the word amino means there's antibodies there. So if we have an antibody to our bait, can it pull down bait and prey? So you're assuming your antibody, which a lot of times can be monoclonal, doesn't bind to the same place the prey handshakes. So you can miss something there. So multiclonal, polyclonal antibodies might be the way to go there for your pull down assays. And then there are others that are physical biochemical approaches, probably you'll get in organic, that we won't really cover here at this point. There is a database, and I don't know if this is still out there, maybe subsumed, but at one point there was a database of interacting proteins. And so that's probably being updated all the time. So if you have domains or proteins, you can check that to find potential others. And I think there's other software that y'all found in your inquiries. Okay, so let's turn to co-immunoprecipitation here. So in this cartoon, we start out with, we have an antibody to the protein X handshakes with Y. And there are other mystery proteins that we really are not interested in. So we're not interested in these. We're interested in this interaction here. And so we have an antibody, And there's something called protein A agarose B. So what that is a lot like the affinity resin, but these agarose beads are very large and have a density and weight to them. It was kind of like an anchor. If you think of molecular anchor, and you're saying, what's protein A? Well, protein A was originally isolated from the Streptococcus bacteria, and it turns out that Streptococcus, when it infects us, actually coats its self with our antibodies, so that evades immunity. So if I'm causing trouble inside the body, what best to cover my cells with antibodies to trick the immune system to think I'm self. And so there's a protein they have found in these bacteria that bind to the constant region of the antibody, this region right there. And so we attach an agarose bead to it, chemically attach, and now this has an antibody. You add that, and it then finds these. You use centrifugal force, and you bring this into a pellet. So this whole complex pellets. These are not dense enough to pellet. So they're in the supernate, and you wash this 3 or 4 times tenderly, no SDS. You maintain the native interaction. And then you do some more heavy interactions by heating or other kinds of high salt, and you break the antibody bonds here, and you then do mass spec or Western, because we know the X, we're trying to find who's the Y, right? Who is the Y protein? So that's co-immunoprecipitation, using immunoprecipitation, which is bringing down a protein with an antibody complex out of solution. So precipitate means falling out of solution, right? It's like if you put too much sugar in iced tea, the sugar reaches the point where there's no more sugar to be dissolved, and the rest of the sugar drops down to the bottom there. I mean, you stir, and yeah, if you heated it up, you could put it in, and you've probably already had that in chemistry, how to make a super solution here. And so here's another cartoon, the principle and pitfalls of detection by co-immunoprecipitation. So we have our cell, and here's our bait and prey, X and Y, and they happen to be in the nucleus. And there are other proteins such as A, and there are soluble proteins such as B. Now, normally B never sees X, Y, or A, right? Because it's doing its business in the cytosol. These are hanging out in the nucleus. If you lyse cells with detergent, okay, lyse cells, and then do immunoprecipitation, sometimes you'll get what you're looking for, but other times you might get this. You actually bring down B, which does not normally interact, but just by chance has enough antibodies that will handshake with X. And so now you pull this and put this into the informatics database, and it would include B. But in reality, it's not B. So there may be some fractionation required to finally get to this point. Or you might have an antibody that will also bind A. So a lot of times when you're testing antibodies for co-immunoprecipitation, you need to be clear that your antibody is specific. If it's not specific, then if you did a Western, you would see multiple bands. That would be an indicator, we've got something, and that's called cross reaction. And just by chance, X and A have the same set, maybe they're cousin proteins, and they have the same amino acids that form an epitope for the antibody. So that antibody would not be of use to us here. And if you didn't pre-fractionate the cells and separate the cytosol and the nucleus, then you would be picking up B. And imagine maybe how many months you might go down the track. Imagine even trying to publish that, right? And so you're saying, well, what would happen? Well, if you had A and you found B, and if you did, say, the yeast 2 hybrid, so say you started with an antibody first and then did the yeast 2 hybrid, and you tested X and B versus X and Y, uh, this interaction would have a strong interaction. This would have hopefully none. And so right away you would see, hey, something's going on here. And 2 hybrid does not confirm this normally, but it does confirm that. So again, we have two methods that kind of double check each other on that. And this is called that means two or more immunoprecipitation here. And so this is your concept. We have beads, or some people actually use magnetic beads. How about that? And use a magnet instead of centrifuge, and you pull the solution out. But in this cartoon, they're just pulling down one. It's called a immunoprecipitation. It's just pulling down one here, and you figure out what that is. So that's the regular traditional immunoprecipitation. And then this is your co-immuno, where you're grabbing X and Y. Here's the triangle and the circle here. And then you can do further analysis. This could be 2D electrophoresis. This could be, after that, spot analysis by Maldi-Tal for 2 hybrid. Electrospray, and you're doing Edmond degradation. You're doing fingerprinting. You're doing techniques to identify what the blue circle is if you have an antibody to the triangle. Okay, so this is a traditional co-immunoprecipitation here, traditional. So this is an example that I have here. And this is using a and it's abbreviated IP. So if you see in the literature co-IP, that's co-immunoprecipitation. And so what they did in this is kind of ingenious. They were, there was a protein called PAC, and they stuck PAC with a flu protein known as HA or PAC PDB, so protein binding domain alone. So they put it in a cell line that is shown as number lane 3. So lane 3, they took the expressed the PAC PDB in cells, okay? And lane 3, they then ran a Western blot, and they did what they suspect binds to PAC is RAC. RAC and PAC are protein partners. So when you do that, you did not, if you look at lane 3, there is no Western blot detection when you pull down just PAC by itself. If, however, you put RAC1 that's been mutated, a Q at position 61 to L, that's actually a dominant constitutive active, so RAC is always on, and its partner PAC, then they do bind each other. So if you purified, co-immunoprecipitated with a high antibody, you pull down RAC and PAC, and you did a Western and you tested with the anti-RAC, there it is. And if you did an anti-Ha, you would find, here's the PAC antibody in lane 2 and in lane 3. But remember, there's no RAC here in lane 3. Lane 4 is a control, okay? And lane 1 is a control using something called GST-Ha. And it pulled down neither PAC or RAC because it's not attached to PAC or RAC. And this is a confirmation that you did a co-immunoprecipitation. And if you you should see both proteins. So in lane 2, we see RAC with its PAC. So they do interact with each other here. But if you just put PAC by itself, it does not bind to RAC in the cell, and there's no RAC there. So go through this experiment, but this is how the advanced use of co-immunoprecipitation. So followed by Western analysis using antibodies to both. This is assuming we suspect that RAC and PAC bind in their antibodies. Remember, there are companies that do have antibodies to a lot of human proteins. So if you did an original co-immunoprecipitation with PAC and you pulled another protein that by Maltitolf you identified as RAC, you could come back, that all rhymes, doesn't it? You could come back with this after you ordered the anti-RAC antibody, and I assume you have anti-PAC antibody because you did a co-immunoprecipitation and you did this exact experiment. And so that's why those companies have a big list of human proteins, because they never know if somebody is going to need that antibody. So they charge you a pretty penny to get that antibody. And remember, up to this point, you were playing with RAC. You had no idea it bound up to, to PAC. You had no idea it bound to RAC. So this can be more of an interaction there. So this is just a list of how people do that Western blot. That's probably an X-ray film. Those bands were probably light induced, and they exposed the X-ray film here, and that just shows you how you look at that. And those bands can be scanned for intensity and the amount of protein there. So what are some controls when we're doing a co-immunoprecipitation? So well-characterized antibodies. We want antibodies that we know exactly what they bind. They don't cross-react. So we're dealing with known quantities here. We want control antibodies from the same species that are antibody to, say, the PAC is. So if that was a goat anti-RAC, then we want goat serum that's never seen PAC, the human PAC before. And that way you could use that in a Western and it would not find then any, reaction there. So there should be nothing in the goat immune system that should react to a human protein unless you inject it in there. Again, we might need polyclonals to the same protein if X and Y, if one covers the antibody epitope, so that would not be good. If we have mutant cell lines that are lacking, say, the PAC protein, that'd be a good cell line to do your control because you can never co-immunoprecipitate PAC in a cell line that doesn't have it, okay? Or cell lines that have a mutant version that your antibody doesn't recognize. And then you change salt, temperature, other kinds of buffering conditions to make sure there's non-specific proteins when you're co-immunoprecipitating. You don't want any non-specific proteins to interact there. So those are known controls in the co-immunoprecipitation. Here is another example looking at cancer cells. And this is renal carcinoma cells. And they have a high epitope tag protein known as VHL. Don't know what that is. And they have this VHL protein that is a mutant. And that mutation, cysteine to whatever the F amino acid is at position 162, what they have found when that mutation occurs, somebody gets a tumor. So this is the cancer version of the protein. So this is an oncogene, and this is the wild type unmutated. This is the cancerous. And they labeled the protein with radioisotope methionine, lysed it and immunoprecipitated with an anti-Ha. And then that co-immunoprecipitated anything that this protein would bind to. So normally in the wild type, what does it bind to? Does it bind to something different when there's a cancer? And so we have mock cells that show background interactions, and this is shown those, and you see nonspecific bands. This band right here is nonspecific. Okay. Yes, I realize there's something there. So when we do wild type, we find this protein normally binds this protein here, and that's been identified by maltitol for fingerprinting, or Edmund, as fibronectin. This protein binds another protein called QUL2 normally. There of course the protein is. And then it also binds two other proteins, the longan B and C as well. And notice each of the specific proteins is not shown in the controls, right? It's not there. It's not there. This is a negative. Okay. Now, what about the cancer version? Notice if you just do one amino acid, now this protein does not bind fibronectin. No, that's probably not good. Does not bind CUL. That's probably not good. And does not bind the elongans, B and C. A lot of those are making cells from the kidney stay in place because they're bound by fibronectin and elongans. So they keep the kidney cells in place to be a functional kidney. Now we have a cancer and those cells are not held anymore. That's why you get metastatic, because the cells are not bound anymore because the mutation has released the protein interactions here. It's a really sweet experiment here showing how just one change here has changed the protein-protein interactions. So certainly gene therapy would try to figure out a way to reestablish this connection here in somebody that had that cancer, that renal cancer. Okay. Just to let you know that there are viruses that will bind to human proteins, and we can use this same kind of precipitation concept to find out what virus proteins bind and who those proteins are. And this is a cartoon where they did a co-immunoprecipitation with an antibody to this little clear protein here. That's a viral protein, a gray protein, and it has X and Y from humans. And we were able to pull down those proteins. And so in control cells, we see areas that are no proteins. And infected cells, we now see that the proteins, 5 different proteins, actually 6, here's X, Y, and these are actually X and Y are now bound. And so we now can deduce what the virus is doing. It's actually pulling in whatever these proteins are in humans and sequestering them. And a lot of times these can be proteins that are involved in transcription or dividing the cell or monitoring the cell regulators. And now this virus has come in and grabbed them, and that's probably not good for us. And so this is a way you actually show that, and that in fact X and Y are bound by the viral protein here. And so if this virus is really a hot virus, then we now could design drugs here that stop this. So we have somebody infected. If we had a drug that would shut this protein binding down, cover it, break this interaction, we might defeat the viral infection. Just an example. And this is a kit from some company, and this was showing the big picture here. This is a co-IP kit that really works. So they show some type of bead with they're binding the X is the pink, and then the Y proteins, the praise here are in blue, and they break those apart. And notice they do the Maltitolf or the charge mass ratio interaction to identify them here. So they're claiming this kit can figure this out very quickly and efficiently. And of course, they call them dyna beads, right? You don't want to call them pora beads. So dynamic, wow, these things really work. So I add my antibody of choice, I incubate, and now these beads are attached. They have antibodies. I put them in some tissue samples that we broke open, homogenized, drop them in, shake them around. Here's 0 minutes. So we let them go for 5 minutes, and everything that the known protein or tag is here, it grabbed all the mysterious unknown proteins in yellow. Those are then, here's a 1D gel that then goes to a 2D gel. The spots can be subjected to Edman degradation or Maltitolf, mass charge, protein sequencing, protein fingerprints. And now we know what our protein is binding to under these conditions, right? So this can be disease, wild type, drug treated, fetal versus adult cells, infected cells, non-infected cells. The variation is in what we're testing. If this works, then we can, okay, here's our profile. What happens when they're drug infected? Or say bacteria viral infected. What's the profile look like? Gives you a big start. And of course, informatically, all these experiments, if they're published, are now characterized. Like, here's this mumps virus, okay, and it, whatever the mumps protein is, binds these. That's in the database now. So anybody studying the mumps now knows that these are potential targets that are facilitating, the virus's effects. So maybe figure out some way to stop that effect. Okay, so I'm going to stop here with the co-immunoprecipitation, and the next technique is called GST pull down assay. So come back and have some fun, and we'll talk some more.